right, and this is again, this is all in Canvas as well. So we are getting our next phylum today. All right, so we'll go ahead and partition this off here. We're looking at the phylum Annelida. A major jump in structure of organisms here. And the general name for this group is the segmented worms. And that's what the Annelida means, basically, is the annuals or the rings that occur in the organism for segmentation. But the key features of this group that we're looking at as a new feature is we have a true coelom. So we're going to define what a true coelom is. Our nematodes and rotifers last time had a pseudocoelom, but the true coelom is a cavity that's entirely lined with mesoderm. And the main feature we have here is that when you have a gut tract lined with mesoderm, you can have things like digestive organs, like stomachs and crops, and all kinds of internal organ structures with mesoderm. The nematodes didn't have. The next feature we have on the organisms is a condition called metamerism, which is really the fancy way to say segmentation. Metamerism is a segmented body plan that's using kind of repeated segments for adaptive features. So that's kind of the two things we're looking at here. We have three classes we're going to look at. Let me write these out here. Class Polychaeta, class Oligochaeta, and the class Hirudinea. Okay, right. So metamerism. What metamerism means is you have repeated segments based on the same design. We kind of saw this with the tapeworm, right? The tapeworm had like repeated segments, but each segment in the tapeworm was like a repeated worm. The tapeworm was kind of like a Pez dispenser, right? Like it has like, you basically, they released individual proglottids which crawled away as an individual worm. But that was kind of an example of serial homology. It was kind of like a colonial flatworm. This is a little different in that it's a whole organism. It's all one organism. But the repeated segments can be slightly modified for different functions. Like on the head segment will have sensory structures like antennae or eyes or feeding structures. And then the front part of the organism has maybe some digestive parts, so a crop and a gizzard and gonads. And then the posterior part of the organism has maybe some intestinal region for absorption. But the idea is each segment kind of has all the same parts. The parts can be modified for different use. So anyway, certainly on the lab exam, on some annelids, I'll ask you what the segmented body plan is called. So metamerism is a fair term to have on the exam. All right, so typically what we got is a segmented organism. It's segmented not only externally but also internally. There's some internal compartmentalization. So let me go ahead and draw an example of what we're talking about here. So here we go. So what we have going on is kind of a segmented body like this. Okay. So we could say we have kind of ectoderm here, kind of layers of tissue on the outside of the ectoderm. And then for the gut tract, green for that, we have some internal organ structures here we'll point out. Okay, so here is our endoderm, would be our gut tract, internal lining. And then there is mesoderm lining both the ectoderm and endodermal layers. So the key feature of a true coelom here is that our body cavity is entirely mesodermally lined. Mesoderm, both covering the outer part of the body and the gut tract. And it's the mesoderm on the gut tract that enables it to have structures like stomachs, etc. So we have mesoderm. And then that's what results in this being a true coelom. All right. So we're going to do a worm dissection today, and so we'll kind of see this internally. But I also have some cross sections to look at. This is a slide of the worm cross section. We have there's earthworm internal cross section here. Slides there. There's also some leeches on there too, but you have to kind of pay attention to which ones you're looking at. If we did a cross section through here though, so look at our cross section, CS. This is kind of what we're looking at for what the cross section here is. So we have our ectoderm here. We have our endoderm. Let me show you kind of how this looks here. Okay, so there's our endoderm. And then we have mesoderm lining not only the outside part of the body cavity like we saw in the nematodes, but the mesoderm is also surrounding the gut tract there. So we have mesoderm. There. Okay, and that's what results in this being a true coelom. Very important feature. So all of the rest of the organisms that we see this semester are true coelom organisms. So it's going to be the common feature for everything for the rest of the semester. Having a true coelom is the thing. Okay. Okay. So we have a cross section here. Here's one feature I want you to check out. Because earthworms are eating their way through the soil is passing through their intestine. They don't have like a coily intestine like you'd think of like a mammal has, because they're basically eating their way through the soil just passes through the intestine. But they do have an increased surface area for absorption of that. And that's this structure here, this little kind of tonsil looking thing is called a typhloid. So I'll mention that again when we do this section. But it's basically, it's an internal increase in surface area of the intestinal tract for digestion. This cavity here is the gut cavity, the gastrointestinal tract cavity. So the food's passing through here. The typhloid is where the nutrients are being absorbed and then transferred into the dorsal blood vessel. And they have a circulatory system that's separate from the coelom system. Okay, so be sure you take a look at the cross section. I'll probably have a cross section of the annelid on the test and ask you about the coelom, ask you about the lining of this internal cavity, which is endoderm, and then possibly ask you about the type of soil as the absorptive region. Okay, so we have three classes of annelids that we're looking at. The polychaetes are all marine, or mostly marine. There's a few freshwater examples, but mostly marine. And the name polychaeta refers to the bristles that occur on the side of the organism. So the organisms that we're looking at here as polychaetes are the clamworms, the feather duster worms, and mudworms. Mudworms, feather duster worm. I think what I have over here is a clamworm and a mudworm. I only have a few specimens of them, so we'll take a look at those. But the main thing we're looking at with the polychaete is they have, as their key features on their body, is they have a parapodia. I don't think what that means. And they have many bristles. I guess parapodia with many bristles. So what they look like here in cross section, a polychaete looks like, is in cross section they have something like this. It looks like ears or flaps on every segment. So these are the parapodia, and the bristles basically are stout bristles that occur on the upper and lower lobes of the parapodia. So what we got going on here, let's see if I can get some labels here. These are parapodia. And the bristles are basically stout hairs that occur on the parapodia. So they use the bristles kind of like track spikes or cleats when they're burrowing through the sand or when they're in the mud, etc. They're essentially kind of like little spiky locomotion. They use them for movement there. And basically every segment has some fleshy lobes. So you can see here's some typical clam worms here. All these little fleshy lobes on the sides of the worm are called parapodia, and on those will be some stout bristles. You can see this is a lugworm. Lugworms are like usually mud burrowers, and then feather duster worms usually have kind of like this big, kind of like a broom or plumage type situation. But the polychaete worms, let me see if I can pull these out here. I have are just these. So these are all in jars. I'll pull some of these out and put them in a dish, the clam worms. But these ones will stay in the things. This is a lugworm. These are clam worms. These are clam worms. I'll pull some out and put them into a dish when we take a break, or when we break the class here. And but I'm kind of running low on my polychaetes. I need to replenish those. Okay, so here's one more. This is a cheap worm. These are all polychaetes. So probably the questions I asked about polychaetes, I'd have maybe one on the test. There you go. Okay. The oligochaetes, these are the earthworms and like tubifex worms. So oligochaetes are earthworms, tubifex worms, like what you feed your fish. The oligochaete has no parapodia and few bristles. So no parapodia, none, And the name oligo means few in Latin. So if you've ever heard of the term like oligarchy, it means a country that's ruled by a few people. So oligo means few in Latin. And these organisms typically have just 2 upper and 2 lower bristle sets. So in an oligochaete, an earthworm cross section, it looks like this here basically, but there's no parapodia. But typically what you'd see is a pair of bristles on the dorsal and ventral lateral sides of each segment. We have some live earthworms here. I'll euthanize some so we can dissect them. But I'll leave some alive, and I encourage you to actually kind of pick one up, and you can feel the bristles if you kind of hold it in your hands and kind of run your hand down the worm. You'll feel when they're moving, they kind of stick these little bristles out they use for like grabbing on traction. So you can feel those bristles in the earthworm, and it's pretty common. So that's what makes an oligochaete. These are all freshwater. Let me move my slide here. These are all freshwater or semi-terrestrial. So that they really aren't truly terrestrial because they need to live in the soil to survive. So freshwater and semi-terrestrials, they live in wet environments basically. But there's no marine oligochaetes at all. Okay, kind of a cool thing is the mating system of these worms. So here's, if you're not familiar with what tubifex worms are, they're like what you feed a fish, aquarium fish with. Little bloodworms. But most of them we're looking at are these large night crawlers or earthworms. The mating system is kind of interesting. If you look at the outside of the body of these oligochaetes, they have this kind of swollen structure here, which is a good test question. So here's the head end. You can usually tell because it's pushing forward, and the head region in front of this structure is shorter. But this this kind of swollen region here, gland, is called a clitellum. And the clitellum function is to secrete a cocoon or an egg wrapper, basically. So what they do, this is kind of like the sex lives of earthworms. It's kind of interesting. They have on their segment 14 and 15, they have a male and a female opening on segment 14 and 15. Segment 14 has the male seminal vesicle opening where the sperm comes out. And segment 15 has the seminal receptacle where the eggs come out. And those occur just in front of this clitellum structure. What they then do is when they're mating, they kind of orient themselves like head to head, like this. So here's the other worm. And these oligochaetes are hermaphroditic, so they have both male and female gonads. But basically, what they do is the male of one worm lines up with the female core of the other worm, and vice versa, and so they exchange sperm. And then what they do after they exchange sperm is they fertilize their eggs, and then they'll dump their eggs out. But the clitellum basically secretes a cocoon around this whole thing. So this cocoon is where they deposit their eggs, but it's the eggs from both worms that have been fertilized by both worms into that kind of, looks like a sleeve or like a sock or something. But anyway, that's what the clitellum function is. And so if you see an earthworm that has a really pronounced clitellum, you know it's fertile and ready to reproduce. If you see an earthworm that doesn't have a clitellum present, it's immature or probably not ready to reproduce. So again, on the test, I'll probably have an earthworm on the exam, ask you about what that structure is called and what it's used for. And then I might ask you about gender, male/female circle, right? So be sure you know that these are actually male and female combined together in the same worm. Okay. So oligochaetes, few chaetae, no parapodia. They're basically freshwater and semi-terrestrial. And then we have the Paraginia, which are the leeches. Leeches are all freshwater and some semi-terrestrial leeches. And the leeches have no parapodia and no bristles. So there's no chaetae, no parapodia, no bristles, and they also have reduced or no internal septi or internal compartments. When we do the earthworm dissection, I'll show you the segments are actually divided internally in earthworms and in polychaetes usually. But in the leeches, they've done away with the internal segmentation. So segmentation is only external. So no septi. And what they do have, though, is they have oral and anal suckers. Okay, so oral and anal suckers. And so a leech basically looks something like this. Usually the head is kind of the tapered end, and the posterior end is kind of more of the chubby end. And the segmentation is superficial; it's all on the surface, not internal. So here's like what a typical leech might look like. So here is the oral sucker, and here is the anal sucker. And they use these for locomotion. They'll kind of anchor themselves with their anal sucker and push their head end out, and then they'll latch on with the oral sucker and then pull their rear end forward. Most leeches are predators. But the ones that we're familiar with are ectoparasites. They're bloodsuckers on fish, reptiles, and mammals. And so here's a cool article if you want to go jump into Canvas on your own Thursday for sure. They're still used medicinally. Some doctors, surgeons will use medicinal leeches because what they do is when they latch onto you, they will bite a hole into your epidermis or basically your upper skin layer, and then they'll secrete or inject a saliva that's an anticoagulant. So it basically breaks up blood clots. So where medicinal leeches are useful is for bruising. Like if you have an injury where you have hematoma, blood vessels have broken under your skin, and you have a bruising, leeches put on that wound can actually break up the bruising and reduce that hematoma, among some other uses of leeches. But they're still used in medicinal purposes. And then, if you've ever seen the movie Stand By Me, you gotta see this scene. So I'll let you go watch it. If you know what I mean, it's pretty funny. But the idea there, or the African Queen would be one where leeches make a prominent scene in a movie. So there's some fun scenes there. Okay, so what this is all about here, and I have some leeches here. Here's my leeches. I'll put them in a little dish. When we break, take a look. That's the only ones I have.
most of the leeches around here, around in California, are predators. They're not parasites. So you have to go to like the south, Mississippi, Louisiana, to get more aggressive parasite style ones. Okay, so what's going on with the segmentation is locomotion. So again, I have some live earthworms here. You can handle these. You can wash your hands or get your hands wet when you handle them. But they're basically using their internal coelom compartments as a hydrostatic skeleton. So this is the first organism that we're seeing that can actually do what's called abduction. So adduction is where you bring your limbs closer to your midline. Like if I bring my limbs closer to the middle, or if I shorten my body, I'm contracting muscles. That's called adduction. Adding meaning you bring it together. Abduction is where you push a limb out or further away. And so far, none of the organisms that we've seen actually have the ability to do abduction until this group. And it's because of this hydrostatic skeleton, this water skeleton, that they use to have opposing muscle forces. So what they do is they squeeze their segments, they constrict them, and that elongates the segment, and that pushes their head forward. And then once they've locked in with their key, they'll use their longitudinal muscles to pull their rear end towards their head. So earthworm locomotion basically is using the hydrostatic skeleton of water pressure inside the coelom function. Okay, so let's take a look at our dissection here. So I'll euthanize these worms in alcohol. It'll take a few minutes to do that. And the easiest way to do this section would be we have these metal pans here. So grab a pan with wax on it, and then we have these super fine point scissors. So make sure that when you get scissors, don't get the big blunt ones. Get these ones with the really, really fine points, and those are the easiest ones to use for the dissection. Anyway, what you're going to do once the worm is dead is you're going to hold it in your hand, and you're going to insert the tip of the scissor just behind the patellum. So kind of go about halfway down the worm behind the patellum. Poke the scissor in, and then lift as you cut, and you're going to snip all the way up to the head of the worm. The easiest way to do this is to hold it in your hands and then snip anteriorly as you go. You want to make sure you're doing the dorsal side, and you can tell dorsal because there's a dorsal blood vessel that runs down top of the worm, and you'll probably be cutting that open, but you're cut dorsal side. Don't cut the ventral side. You won't see anything of interest in the ventral side. So put the scissors in, cut all the way to the tip, and then once you get it opened up, you can put it in the pan and use pins to kind of pin it out. And we have some T-pins at various places around the room here. We have T-pins like this that you can use to kind of open the worm up with the pan. These days with cell phones, it doesn't really matter where you put it in the pan, but if you end up on the exam, I'll use one of these dissecting microscopes to show. And unfortunately, the dissecting microscopes, if you put the worm right in the middle, you won't be able to see anything. So I'd recommend when you pin the worm, pin it off like to the side. Pin it off like maybe a quarter of the way to the edge. And that way, if we do use a dissecting microscope to look at the structures, you can kind of get in there and move it around and see things. But these days, the cell phones are so good that they magnify and have sharper images than pretty much almost our dissecting scopes do. So I think you can see everything you need to see by using your cell phone, zooming in on the structures. Okay, so let me go ahead and the cross-section picture there basically is showing what you need to see in your cross-section. There's more in the cross-section than what I'm going to ask you. Basically, I'm going to limit it to coelom and typhlosoil. It's kind of the main thing I'm interested in. And the coelom is of note because it's entirely laminotestular. It's kind of the question there. But with the earthworm dissection, there's more going on there. And so let's go ahead and draw that. So what you're going to end up with basically is you're going to have the worm's head here, and you will then kind of butterfly the body like this. Here's kind of a worm like this. So you basically made a cut that ended at the head and started about halfway down like this. So you've snipped here, cut all the way to the head, and then you've kind of teased the body out like this. So okay, let me go ahead and I'm going to start off with looking at the digestive structures. So here's kind of like the, online image you'd see, but I think it's probably better if I'll show you exactly what I want you to know on this for the exam. So let's start with digestion. So there is essentially 4 intestinal organs that are important. One is very, very head end. There is a pharynx. Pharynx is kind of at the very, very head end. There is an esophagus. I'm not going to label it because you won't be able to see it. The esophagus is covered with other stuff. But this tube here is an esophagus. And then there is a pair of stomachs here. One is called the crop, and the other one's called the gizzard. So the crop is a soft stomach. If you poke it with a pin or with your probe or what have you, it kind of gives, and you can feel that it's kind of like a baggy, or it's soft, meaning it's membranous. The gizzard is muscular, so when you poke the gizzard, it actually kind of pushes back and doesn't really fold up. So what's going on is the crop is like an enzyme stomach that dumps enzymes onto the food, and the gizzard is a grinding stomach that uses kind of muscles to kind of grind up the food. Then the intestine is in the posterior portion, and the intestine has in it a typhlosil. And you can see this typhlosil. It's basically a blood vessel-rich structure. If you accidentally snip the intestine or intentionally, you'll actually see this kind of reddish-orange thing flop out. And that reddish-orange thing basically is this typhlosil here. And it's the blood vessels that are in the typhlosil that are kind of popping out. And it looks like ribbons, like an orange ribbon coming out of the intestine. So be sure you check the intestine out and see if you can find the typhlosil. And certainly I'm going to ask you about typhlosil on the cross section, so be sure you see that. Okay, so that's basically the digestive organs that you have to know. The esophagus, like I said, is covered up by a wide variety of things. When you first open the worm, it's covered with mostly seminal vesicles and seminal receptacles. There's a whole bunch of these big pillowy looking things on top of the esophagus. I'm not going to ask you any of the sex parts of these worms, but basically the big pillows on top are sperm storage organs and sperm transfer organs. But the main thing I want you to see is the circulatory system. There are 5 pairs of hearts or aortic arches. 4, 5. So 5 pairs of what are called aortic arches. Some textbooks call these hearts. They're not technically hearts because they're inline pumps. They're kind of inline muscular pumps that pump the blood around the body. But take a look at those. When these are really fresh, you can actually still see them kind of beating or circulating or doing their thing. The 5 pairs of aortic arches are important there. And then I do want you to see, these are important structures. Let me see if I get a good color here. There is, you'll find, that's kind of hard to see. Use paintbrush. No. Limited my color. Orange. Try orange. Okay. There are 2 pairs of what are called calciferous glands. Calciferous glands. The calciferous glands, there's 2 pairs of them, and they're actually behind arch 4 and 5. Aortic arch 4 and 5. The calciferous glands are used for regulating blood pH. What the calciferous glands do is they absorb calcium from the intestine as the food's passing through the esophagus, and then transfers the calcium to the blood. And the reason for this is earthworms live in the soil, and typically soil is acidic. Soil is usually kind of like pH 5 to less than 7. So acidic soils are more common than basic soils. The problem with having hemoglobin is a blood protein that carries oxygen, is in acidic environments, CO2 builds up too much. So the calciferous glands are for maintaining blood pH at near 7 or near neutral. And so earthworms need to be able to bring in calcium in order to make their blood basically pH neutral, so they can carry oxygen. So if you grow worms, like if you have a worm bin or a worm farm, many people will do things like put chicken eggshells into a worm bin, and the worm will eat the chicken eggshells and transfer the calcium. Or they'll spread calcium dust in the worm bin because that benefits the worm. But anyway, the calciferous glands are a blood pH gland that basically is taking calcium out of the food and putting it into the blood. All right, lastly, I mentioned that there is these internal septi. So the inside of the body basically is divided into compartments. And so you'll see this, when you tear it open, you'll probably tear a lot of septi, but take a look at like the very back of your cut here. You'll still see some septi intact. And I'll probably ask you about septi on the section of the worm. They're pretty obvious. But the thing that's totally cool to see if you can see is the osmoregulation organ of the worm. And this is what's called the nephridia. So the nephridia are like a hairpin loop structure that occurs in every segment. So these are nephridia. I won't ask nephridia on the test because it's super hard to get them reliably seen or exposed, and it's certainly really hard to put a pin in them so I can ask you questions about them. But I want you to see them because probably in about 2-3 weeks we'll cover how nephridia function and what nephridia are. And they're really an amazing osmoregulatory organ. It's an organ that basically filters the coelom fluid and will either retain or get rid of water as needed. So it's kind of like a little mini kidney. And in fact, your kidneys are made up of thousands of nephridia, or nephrotubules, that are probably homologous to these nephridia. So it's a really common way for animals to do osmoregulation and get rid of nitrogenous waste, is to use nephridia. And so this is our first example seeing that. I'm only drawing them in a few segments here, but they basically occur in every segment of the body as hair of nephridia. And in fact, in the cross section, sometimes you can see like a little remnant of a loop like this
and that would be the nephridia that you see in the ceiling. But see if you can find those. I won't ask them on a test because they're hard to identify during a 1 hour exam, keep them visible. But I think they're really cool, and they're kind of transparent. They look almost like a little hairpin, like a little clear hairpin. But you can usually see them kind of here right where you initiate your cut. Okay. All right. So it looks like I need to go get some. Oh, I may have it. I may have to go get some more alcohol here. So yes, yeah, I'll do that. All right, so here's our worms here. What I need to do is I need to go get some more ethanol. I don't seem like I have enough here to euthanize all the worms we need. And then I'll come back with the worms euthanized. You can take a look at the specimens. We have the slides here. We don't need these slides, these are okay. So don't worry about these. We'll just look at the earthworm cross section. There's also some slides here of a leech, a whole mount leech. I won't have these on the exam. I'll probably have those leeches on the exam, but that's what that is. It's basically a whole mount leech. You can kind of see those. Okay, I'm gonna go get some more ethanol to euthanize the worms. You guys can get your dissection cans and get ready to go, and I'll be right back. Questions? Look good? Okay, be right back.
Sweet little pan paper. Maybe a little worms.